Hormones can be classified according to their chemical structure. The synthesis, secretion, and transport of hormones depend on the chemical properties associated with structure. The control of secretion of a hormone depends on its function. Your goals for learning are to recognize the chemical classes and solubility properties of hormones, to observe how a single member of each class of hormones is synthesized, to know that secretion of hormones depends on release, synthesis, or both, to learn that neural, hormonal, or humoral stimuli control secretion of hormones. Here's what you need to know. The structures of amino acids, proteins, and steroids. The difference between water-soluble and lipid-soluble. The concept of negative feedback systems and how they work. The synthesis of complex molecules proceeds along pathways, each step requiring a specific enzyme. The anatomy and neurotransmitters of the autonomic nervous system. To see definitions of terms, click the bold red words. Recall that a hormone is a chemical messenger that is released into the bloodstream. Most hormones can be grouped into one of three classes of chemicals: peptides, amines, and steroids. Click the word peptides. Hormones made from chains of amino acids range in size from small peptides of just 3 amino acids to small proteins containing more than 20 amino acids. For simplicity, we will refer to all of them as peptides, a convention used by endocrinologists. Most hormones are peptides. They are water-soluble. We will study synthesis of the peptide insulin in detail in the next pages. Click the word amines. Amine hormones are derived from the amino acid tyrosine. Dopamine, norepinephrine, and epinephrine are called catecholamines. They are formed by a series of chemical reactions that modify tyrosine in successive steps. Catecholamines are water-soluble. We will study the synthesis of epinephrine in detail in the next pages. 2 iodine-containing molecules, thyroxine, or T4, and triiodothyronine, or T3, jointly form the thyroid hormone. Each is produced as iodine is added to thyroglobulin. T4 and T3 are lipid-soluble. We will study the synthesis of thyroid hormone in detail in the next pages. Click the word steroids. Steroid hormones are derived from the molecule cholesterol. Steroids are formed by a series of chemical reactions that modify cholesterol in successive steps. Steroids are lipid-soluble. We will study synthesis of the steroid cortisol in detail in the next pages. We will now study synthesis of the peptide insulin. Recall that insulin is produced by beta cells in the pancreas. Click the rough endoplasmic reticulum to begin. Peptide hormones are synthesized on ribosomes often as large precursor molecules called preprohormones. Many are processed in the endoplasmic reticulum into smaller peptides called prohormones. Click a preprohormone in the rough ER to see the prohormone for insulin. The Golgi apparatus packages the prohormones with other proteins into secretory vesicles that are stored within the cell. For some hormones, conversion to the active form takes place in the vesicles. Click a prohormone in the secretory vesicle to see conversion to the hormone insulin. Drag a secretory vesicle to the plasma membrane. In response to stimulation, secretory vesicles move to the plasma membrane and fuse with it. As they open, their peptide hormones, along with cleaved fragments and other stored proteins, are released into the extracellular space by exocytosis. Stored hormones can be released rapidly into the circulatory system. Endocrine tissues are well vascularized so that hormones can easily enter the bloodstream for transport to target cells. Since they are water-soluble, peptides are carried in the blood plasma as dissolved particles. To see the entire sequence, click the rough endoplasmic reticulum. The synthesis and storage of amine hormones varies for each hormone. Recall that epinephrine is produced in the adrenal medulla of the adrenal gland. Click the molecule to begin the study of epinephrine. Cells of the adrenal medulla are modified postganglionic neurons of the sympathetic nervous system. Like postganglionic neurons, they are innervated by preganglionic sympathetic fibers and they produce catecholamines. Unlike postganglionic neurons, the adrenal medullary cells lack axons and secrete their products into the bloodstream for dispersal. In most postganglionic neurons, norepinephrine is the primary catecholamine and it functions as a neurotransmitter. In adrenal medullary cells, epinephrine is the primary catecholamine. Because it enters the bloodstream, it functions as a hormone. Click the molecule to continue. Synthesis of epinephrine begins as tyrosine is converted to dopamine. This conversion takes place in the cytosol where the enzymes for each step are located. Dopamine is pumped into secretory vesicles called chromaffin granules, where the enzyme for conversion to norepinephrine resides. Click the secretory vesicle to continue. Both norepinephrine and epinephrine are produced by the adrenal medulla in a ratio of 1:4 or 1:5. Production of epinephrine requires norepinephrine to re-enter the cytosol, where it is converted to epinephrine. It is then pumped back into the vesicle. Secretory vesicles serve two purposes. They amass quantities of catecholamines for rapid release, and they protect norepinephrine and epinephrine from breakdown by cytosolic enzymes. Click the secretory vesicle to continue. Hormones are released from adrenal medullary cells by the same process that causes release of neurotransmitters from neurons. Stimulation of both endocrine cells and neurons causes an influx of calcium that induces movement of the secretory vesicles to the plasma membrane. They open and release their contents into the extracellular space by exocytosis. Since they are water-soluble, catecholamines are carried in the blood plasma as dissolved particles. To see the entire sequence, click a secretory vesicle. Recall that the thyroid gland produces thyroid hormone. The mechanism of production of this amine hormone is unique in the body. Click a molecule to begin the study of TH. Recall that thyroid hormone is really two molecules, T3 and T4, produced by special follicle cells that surround spherical structures called follicles in the thyroid gland. Production begins as follicle cells synthesize a protein called thyroglobulin and secrete it into the follicle. The protein contains multiple copies of the amino acid tyrosine. Click the thyroglobulin to continue. As thyroglobulin enters the follicle, each tyrosine binds one or two iodine atoms. The iodinated forms of tyrosine couple, producing either a hormone with 3 iodine atoms, T3, or a hormone with 4 iodine atoms, T4, that is still a part of thyroglobulin. This molecule is stored until the follicle cells are stimulated to release thyroid hormone. Click the storage molecules to continue. At that time, the storage molecules re-enter the follicle cells by endocytosis. The endocytotic vesicles fuse with lysosomes, whose enzymes break down thyroglobulin, thus releasing T3 or T4 into the cytosol. The lipid-soluble T3 and T4, in a ratio of about 1 to 20, diffuse out of the follicle cells and enter the bloodstream, where they bind to 2 kinds of carrier proteins: specialized carriers that bind only thyroid hormones, and the plasma protein albumin. Albumin non-selectively carries many lipid-soluble hormones. The unusual production and storage of thyroid hormone has special consequences. Follicles are able to store 2 or 3 months' worth of thyroid hormone, an important property of an essential hormone that affects almost all tissues in the body. The synthesis of steroids from cholesterol creates a large family of related hormones. The type of enzymes present in a given cell determine which steroid or steroids that cell produces. The metabolic pathways are very complicated and contain more intermediaries and loops than are illustrated here. Click cortisol to begin. Recall that cortisol is produced by the inner portion of the adrenal cortex. Adrenal cortical cells store a modified form of cholesterol in lipid droplets. Synthesis begins as cholesterol is released from storage. The critical step in converting cholesterol to any steroid hormone is production of the common precursor molecule, pregnenolone. Enzymes for the production of pregnenolone reside on the inner mitochondrial membrane. Cholesterol enters the mitochondrion where it is converted to pregnenolone. The pregnenolone molecule must re-enter the cytosol for conversion to intermediaries on the pathway to cortisol production. Intermediaries re-enter the mitochondrion where they are converted to cortisol. Click the cortisol in the mitochondrion to continue. Cortisol enters the bloodstream by diffusion. The lipid-soluble cortisol and other steroids bind to 2 kinds of carrier proteins: specialized carriers that bind particular steroid hormones, and the plasma protein albumin. Remember that albumin non-selectively carries many lipid-soluble hormones. Steroid hormones are not stored, they are synthesized on demand. Therefore, the secretion of steroids is immediate, and its rate depends on the rate of synthesis. To see the entire sequence again, click on the cholesterol. We have learned that the availability of hormones depend on their secretion. Stimuli may alter synthesis, release, or both processes to change plasma levels of a hormone. 3 types of stimuli control secretion of most hormones: neural stimuli, hormonal stimuli, and humoral stimuli. Click neural stimuli to begin your study. Some endocrine cells are directly stimulated by nerve fibers, commonly fibers of the autonomic nervous system. Catecholamine secretion from the adrenal medulla is controlled by sympathetic fibers. We will study the secretion of epinephrine in detail. Preganglionic sympathetic fibers innervate the endocrine cells of the adrenal medulla. When they are stimulated, the neurotransmitter acetylcholine is released from their axon terminals. Acetylcholine causes adrenal medullary cells to release epinephrine and norepinephrine into the bloodstream. The hormones, epinephrine and norepinephrine, reinforce the actions of the sympathetic nervous system. Any stimulus that elicits a sympathetic discharge, stressors such as pain, fear, physical trauma, infection, prolonged cold, or emotional stress, causes secretion of epinephrine and norepinephrine. Click hormonal stimuli to continue. Secretion of many hormones is controlled by other hormones. The ventral hypothalamic hormones and tropic hormones from the anterior pituitary, thyroid-stimulating hormone, adrenocorticotropic hormone, follicle-stimulating hormone, and luteinizing hormone are classic examples. We will study the secretion of thyroid hormone and cortisol in detail. Thyroid-stimulating hormone is the stimulus for secretion of T3 and T4. Increased amounts of thyroid-stimulating hormone act on follicle cells of the thyroid gland to cause more thyroglobulin to be brought into the cell where it is cleaved to release T3 and T4. T3 and T4 diffuse into the bloodstream. The primary mechanism for keeping blood levels of thyroid hormone relatively constant is a negative feedback loop in which free T3 and T4 in plasma inhibit secretion of thyroid-stimulating hormone. Recall that stimulus for secretion of thyroid-stimulating hormone comes from the ventral hypothalamic hormone, thyrotropin-releasing hormone. It is released in pulses that vary in a circadian rhythm. Adrenocorticotropic hormone is the stimulus for secretion of cortisol. Increased amounts of adrenocorticotropic hormone act on cells of the inner adrenal cortex to cause cholesterol to enter mitochondria and synthesis of cortisol to increase. Cortisol diffuses into the bloodstream. Elevation of plasma cortisol is dampened by a negative feedback loop in which cortisol inhibits both adrenocorticotropic hormone and corticotropin-releasing hormone, the ventral hypothalamic hormone that causes secretion of adrenocorticotropic hormone. Corticotropin-releasing hormone secretion follows a circadian rhythm that causes daily variability of cortisol in the absence of stress. Stressors like those that elicit secretion of epinephrine, pain, fear, physical trauma, infection, prolonged cold or emotional stresses also elicit secretion of corticotropin-releasing hormone. Click Humoral Stimuli to continue. Humoral stimuli include the concentration of ions and nutrients in plasma. The function of some hormones is to regulate these plasma variables. For example, insulin and glucagon regulate plasma glucose, parathyroid hormone and calcitonin regulate plasma calcium, and aldosterone regulates plasma potassium. The secretion of each of these hormones is controlled primarily by changes in the regulated variable. We will study the secretion of insulin in detail. The primary factor that controls secretion of insulin is plasma glucose. As plasma glucose rises after a meal, pancreatic beta cells secrete more insulin. As we have seen in our endocrine review, insulin acts on muscle, adipose, and liver cells to promote uptake of glucose and storage of fuel. This causes blood glucose levels to return to their pre-feasting values. Increasing plasma glucose levels initiate a negative feedback loop that causes glucose to return to the pre-feasting levels. This removes the stimulus for secretion of insulin. Note from your study of this page that the secretion of most hormones is controlled by negative feedback loops. They serve to keep hormone levels within a desired range. Eating a meal is the stimulus that initiates the rise in plasma glucose. Although blood glucose is the primary factor controlling insulin secretion, other factors also play a role. They include humoral factors like plasma concentrations of amino acids, hormonal factors like glucose-dependent insulinotropic peptide, GIP, and activity of the autonomic nervous system. Click on the pancreas to continue. When blood glucose is adequate, an increase in plasma concentration of amino acids stimulates pancreatic beta cells to secrete more insulin. Insulin increases the uptake of amino acids into muscle and other cells. This causes plasma amino acid levels to return to their pre-feasting values. Increasing plasma amino acid levels initiate a negative feedback loop, like that caused by increasing plasma glucose. This removes the stimulus for secretion of insulin. Eating a meal is the stimulus that initiates the rise in plasma amino acids. The hormone glucose-dependent insulinotropic peptide, or GIP, causes secretion of insulin. Click on the pancreas to continue. As a meal is eaten, the hormone GIP is secreted from the gut and stimulates pancreatic beta cells to secrete insulin. This is a feed-forward mechanism that causes an earlier and larger secretion of insulin than would occur if glucose were the only stimulus for insulin secretion. Both parasympathetic and sympathetic fibers innervate cells in the pancreatic islets. Parasympathetic activity increases insulin secretion, and sympathetic activity, or the hormone epinephrine, decreases insulin secretion. Parasympathetic fibers become active in preparation for and during eating. Like the hormone GIP, this stimulus is also a feed-forward mechanism that causes an earlier and larger secretion of insulin than would occur if glucose were the only stimulus for secretion. Eating a meal is the ultimate stimulus for both of these feed-forward mechanisms. Sympathetic fibers become active in response to stress. Regulating the supply of plasma glucose is a very important part of the stress response when immediate physical action may be needed. Food consumption and stress are events that influence the levels of plasma glucose. Blood levels of a given hormone can vary widely over the course of a day. Normally, blood levels are determined by secretion. Hormones are not secreted at a constant rate. They are secreted in response to stimuli. We noted that cortisol and thyroid hormone are secreted in response to hypothalamic and pituitary hormones. Epinephrine is secreted in response to sympathetic stimulation, and insulin in response to plasma glucose levels. We learned that secretion of cortisol and TH are modulated by negative feedback loops that keep blood levels within a given range around a set point. Negative feedback control does not imply constant levels of a hormone. We will look at the patterns of secretion of cortisol and TH to illustrate several concepts in control of blood levels of hormones. Click the adrenal gland to continue. Many hormones exhibit rhythmic patterns of secretion. A common pattern is the circadian rhythm, where hormone levels rise and fall in a regular pattern lasting about 24 hours. Cortisol, driven by CRH, has a built-in circadian rhythm that is synchronized with the 24-hour day by exposure to light. For people who are active during the day, cortisol levels are highest in the early morning hours before arising and lowest in the evening. Circadian rhythms arise from the activity of biological clocks in the CNS. They alter set point for a hormone, and negative feedback systems maintain hormone levels around the established set points for a particular time of day. Hormonal rhythms can be shorter or longer than a day, and they serve as feed-forward mechanisms. They allow the body to anticipate changes and prepare for them. Stressful stimuli can be superimposed on this cycle to elicit additional secretion of cortisol. Such stimuli override the set point. Click Continue to proceed. Here's a summary of what we've covered. The chemical structure of a hormone determines its solubility properties. The synthesis of hormones involves shuttling precursor molecules from organelle to organelle or cytosol often multiple times. Families of related hormones are produced when the metabolic pathway involves sequential modification of a basic starting molecule. The hormone produced by a given cell depends on the enzymatic makeup of that cell. Hormones that are not soluble in water are transported on carrier proteins. Blood levels of most hormones vary widely throughout the day. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the